morning, everyone. We've been looking forward, I think, to this session, which is all about solutions. And we all want to hear what the progress is because we desperately need some tools in the toolbox. So strap yourselves in. This is going to be a quick round. We're going to have 7 speakers. We're going to have 3 before our tea, and then we're going to have 4 after our tea. I'm not sure why they put the tea in between. I think it might be afraid of my timekeeping abilities, and I feel under pressure because everybody's keeping to time. But I am known to go on a bit of a tangent, as I am doing right now. But I'll stop and I'll bring up Tom Breen, I think, from Ruminant Biotech. We've got Colin South from Arkea Bio, and we've got David Aitken from Lucidome Bio. So welcome you to the stage. And to keep time moving, Tom, I'm going to get you to take the stage. Look to your timekeepers who are doing a fantastic job. I've been told if you go over your time, I need to take you off. I just let you know, I have grown up with 6 brothers, so I have multiple ways of getting you off the stage. Yeah.
Well, I've grown up with 3 brothers, and I'm pretty good at fighting back. So we'll see how we.
Challenging. Don't light that candle.
Thanks very much. A tough act to follow. That was a great session this morning on the back of a great day yesterday. So I'm really looking forward to give a bit of an update from Ruminant Biotech's perspective. I think we are a long way down the path to solving a few of those challenges that were raised this morning. So Ruminant Biotech, founded in 2021 here in New Zealand by a couple of very visionary, long-standing stalwarts of the industry. They were looking at the promise of methane inhibitors, particularly through feed additives. And they instinctively realised that wasn't going to work for pasture-based systems. If it was going to work, we're going to have to be able to disconnect the delivery of the inhibitor from the feed. And so, with working with animals and boluses for many, many years, they came up with the idea and the concept to get the inhibitor into a bolus and see if you could get a long duration of efficacy with that concept. And so our team has been spending the last 3 years really trying to prove that out, and we're really pleased with the progress that we're making, so I'll take you through today. Firstly, I joined the company in April this year, moved over to New Zealand from Australia. I've spent the majority of my career, firstly as a producer, mostly producing cotton in northern New South Wales, but also beef. I grew up on a mixed sheep and cattle farm in central west New South Wales. But I've spent the bulk of my career commercialising technology for GM technology for Monsanto in Australia and also across the Asia Pacific. So I enjoyed hearing about the conversation, the discourse on GM this morning. I think that is going to play a future role, and I'm glad we're having a really mature conversation about that. And I think a lot of lessons have been learnt from the GM industry around how we take new technology to both farmers, but also how we bring the consumer along with technologies like that inherently sort of raise some concerns when they hear about them for the first time. And I'm really excited that the lessons that we've learned over the last 20 years in those areas, we're going to be able to apply to our space and make sure that we can have the impact we want for the climate, but also make sure that the customer, the consumer and the farmer comes along on the journey with us. First thing about our product and what it is, just 4 key components around the bolus for those of you that are not familiar with it. Number 1 is a densifier. It's just a weight that keeps the bolus in the animal for the treatment period that we want. Number 2 is a biodegradable casing. So we've designed the bolus to degrade over time in the rumen. Once it's released its active ingredient, it can break down then and be cleared from the animal. The formulation, obviously, we use a proprietary blend of bromoform with a carrier formulation that allows us to stabilise the active and then pay out over an extended period of time. And we have an RFID chip component to the bolus as well, which we think is really, really critical from a methane verification perspective. It allows us to know what animals are under treatment, and we think it's going to be really, really important to verify those methane emissions reductions, which are going to be critical for monetisation. So the key advantages of the product, precision dosing, sustained performance. I'm going to take you to a trial of ours that was completed this year where we're pretty confident now that we're seeing up to 75% daily inhibition over a 100-day period with our late stage prototype that we're looking to commercialise. I've spoken about compliance. Most importantly, I think for everyone in this room from a New Zealand context, is it's going to be broadly applicable. So we are going to be able to target animals in pasture-based systems, not just here in New Zealand, but right around the world. We think it's safe. The synthetic bromoform that we use is free from contaminants, and the work is being done to show that it's safe within the animal. And we think it's going to be a very convenient, what I call the phrase, a low intervention activity at the farm. So with a farmer needing to potentially once or twice per year to treat the animals with the bolus, that is a low intervention animal husbandry technique, and we think it's going to be widely adoptable because of that. So in summary, when we put all those things together, we think that's going to be a really, really high impact solution for the industry. High efficacy, sustained duration of efficacy, pretty low intervention at the farm, and easily verifiable. Those things should add up to being a really compelling solution. So just a little bit on the study for this year. So this was, we call this a characterisation of efficacy study. What we were looking for, we had 2 of our late stage prototypes in this trial with a control, 10 animals in each treatment. I'm only showing one of the treatments here, which is the one that had the longest duration of efficacy and the highest overall inhibition. The other prototype had a different sort of results to this. Both of them had pros and cons, and our final generation 1 prototype will probably be a combination of those 2 things. Treated 400 kilo Kiwi cross dairy heifers in the Waikato at dosing, and the leading commercial prototype demonstrated 100 days of efficacy with 75 or 70 plus methane inhibition per day. We had no treatment-related adverse events out of this. We had no evidence of adaptation either. So when you see the back end of this curve where you see methane inhibition start to drop away, we know that's from boluses starting to degrade in the animals and be cleared. So that's good news. It's not about adaptation. It's more about the treatment effects coming to an end because of the physical characteristics of the bolus. So very, very exciting for us. It gives us enough confidence now to think that we've definitely still got some work to do to finalise on our product that we're going to take to market next year. But we are able now to move on from some of the technical work that we've been doing to more of the commercialisation work that needs to be done. So to get to a target launch in October 2025 in Australia, we're hopeful and optimistic that we might be able to have a limited commercial release here in New Zealand as well. We're working really closely with the MPI on that, and time will tell if we can get there. But for Australia, at least, what we're going to have is a product that's got at least 3 months duration of efficacy, 75% daily methane inhibition, a biodegradable bolus, which is going to be important, and a single dosing step per treatment. So a farmer's not going to need to dose the animal twice for a single treatment. What we need to do to make that happen is, first of all, we need to get our manufacturing capacity online, and we're currently in the process of building a facility here in New Zealand. That'll come online from around about March, April next year, and then a couple of months of commissioning after that, which is going to allow us to have some commercial product available from October next year. It'll also allow us to define our manufacturing process, which we can start to take and apply to ramping up capacity around the world. From a regulatory FTO perspective, so we have got the ability to take the product to Australia from October. We've submitted our dossier in New Zealand in September this year, which was fantastic, a huge milestone for the company. It's enabled the Codex MRL setting process to begin, which is critical, and we'll continue to work away with the regulator here in New Zealand on all the confirmatory data that needs to go in to get that full regulatory approval. And then the last piece, and the biggest piece, a lot was spoken about this morning around adoptability. So that's both at the farm but also within the value chain. So really, really important that we can now take our product into a market in Australia. We're going to spend, we've got a commercial market development under trial in Orange, New South Wales at the moment. That's allowing us to build a lot deeper engagement with producers and the value chain to give them an understanding of our business, of the product and the opportunity and the commercials that are going to sit around that. We'll continue to scale that program up over the next 12 months with more trials and larger trials. That'll help us expose the product to a little bit more variability as well across the 4 seasons in Australia and some different management systems and pasture types and animal types, etc. It'll also allow us to get started on the verification work that's going to be needed for farmers to be able to claim their emissions reductions and create value from it. And most importantly, to set up the commercial the commercial framework that's going to allow our product to get to market. We're a small company of 20 odd people. We're going to need partners and a lot of support to make this promising technology a commercial reality. So thank you very much for the opportunity to give an update this morning. We're really excited about where we're up to and the impact that we can have with our methane inhibiting bolus. Thank you.
Thank you, Tom. I'll hand the stage over to Colin South from Arkea Bio now.
I'm going to stop coming to these conferences. I think we need a new rule in that the questions are out there and the solutions are in here, because this is where the venture capitalists sit. And it's not really good look for me to hear 2035 as a timeline. There's a saying that vaccines are 10 years, for methane are 10 years away and always will be. And Harry just pushed it out to 11. That's tough. So, it is a tough story to tell. But some things have changed over the last 25 years since vaccines were first talked about. Markets are beginning to be there. There's a lot of talk about who's going to pay, but someone's going to put this in and it's going to have to happen. And the second thing that's changed is the technology is phenomenally different now. The ability to do large-scale microbiome sequencing, the ability to look at what's going on in real time in cattle, and in fact, the other piece that's really important is why we're here, is that venture is starting to look at this and put money towards it. And so it's a different world than it has been. There's different tools that can be applied. So we're really bullish about the opportunities in vaccines. We've originated with Breakthrough Energy Ventures in the US. Bill Gates founded that with about 30 of his favorite billionaires. And they've got 3 funds now. They've got 3 funds. Each of them's $1 billion fund. And we're invested in out of that second fund. It's really interesting. There's a $3 billion fund there, but it's not actually the behemoth in the space, in that AgriZero, with their focus directly on the problems that are needed for pastoral farming, and as Vinod Khosla would say, it's not a solution unless it works in Chindia. If it doesn't work for China and India, it's not a climate solution. And it just happens that those things align well with where we need to be for a New Zealand solution. We need to be in somewhere that works in low-touch environments. We need to have longevity of action. We need to fit in with existing agronomic practice, and we have to be consumer acceptable. And the vaccine really checks all those boxes in a way that other things don't. I mean, we looked at Bovia problems in the last week. Every carton of milk in the US has no RBST written on it. And sure, there's none. There's none in anything. It never got acceptance because once it lost that battle, it completely lost that battle. So we have to be careful to make sure that consumer acceptance is brought in along with producer acceptance. We were funded in 21. We got into the lab in early 23 in terms of, we got our seed funding at the end of 2022. We were in the labs in Boston, staffed up with about 10 people at that stage in early 23. So we've been in the clinic and in animals virtually through that whole time. We're based in Boston. It's a great place to do biotechnology. You can hire, you can get the tools you need. But in addition, we're partnering with Texas A&M with our cattle work down there. So we've been doing in vivo studies for that entire time, about 18 months now. And so we're excited about where we can get to. It's obvious why we're doing it. This is for the uninitiated, but the amount of methane emitted through cattle is the equivalent of all passenger vehicles or all concrete, or 4 times that of airline emissions. So it's just a huge size of problem, it's a huge size of prize, and we kind of lost the plot in the agricultural sector a long time ago in that we are a solution here. Everyone looks at it and says it's a major issue. This is the most readily addressable large-scale carbon problem that's out there. We can make a change in methane for both carbon dioxide equivalents and also for short-term warming in a way that nothing else apart from petrochemicals stopping their leaks does. Its mode of action is relatively simple. We don't have a big problem. We've just got to get the costs down. We've got to get access to where the bugs are. We've got to have stability in the rumen. We've got to have titer that's sufficient to knock down the majority of the methanogens. We've got to have longevity, and we've got to have efficacy. And we're going to do that, technical ask, at the same time as we're developing new carbon markets and we're selling things to farmers. And in the middle of it sits a regulatory problem. And that regulatory piece is one of the reasons we're down here. This is where the urgency exists in it. The unique situation New Zealand finds itself in gives us a unique sense of urgency. We're looking at very closely at the way in which the regulatory environment in New Zealand and Australia is changing, because it's likely to be one of those places where we can actually move quickly. And moving quickly matters to get that first product into market, but it's also how you handle the incremental products beyond that. We're not a single product like a bromoform or a 3-nop. We're going to have to evolve the efficacy with additional antigens and additional performance all the way through. So they're the kind of the landscape we find ourselves in. Great work that's been done before, and Harry has seen so much of this through over his time. And if we look at the 5,000 genomes project that was done, you see something that's really hopeful for anyone in this space, in that the rumen behaves more like an independent organ inside the animal in terms of just the breakdown of the feed of the microbiome is very similar across species, across production systems, and across regions. So we're hopeful we can get there. That clearly leads us to get a look at the microbiome very closely to see that it's in a very specific niche, and it's directly addressable because of that. So we're bringing new tools to the table here. We're bringing metagenomics, we're bringing our own proprietary databases, proteomics, protein design, and high-throughput screening in a way that hasn't been there before. And that really gives us a toolkit that takes us down from the 10 million genes there are in the rumen all the way down to about 8,000 targets that we've got. And then we address those, and we're addressing the antigen discovery part of which is by far the hardest piece of the journey. And where we are to date, as I said, we've been in the lab about 21 months now. And so we have what we think is the first reproducible data that shows that addition of a vaccine can have a measurable impact on the amount of methane that's being produced. It's not as high a efficacy as we want yet. It doesn't have the longevity, but we're on that path and believe that there's a lot of good reasons for optimism. We see no change in daily gain. We see the antibody response having some longevity out to give us hope to get to where we want to. And from that, we think there is a clear path forward that we can go down here in terms of finding a way to get a vaccine. Hopefully, we're not 10 years off, but there is definitely the clear path that we can see that the methodologies we're using are taking us down to where we want to be. Thank you for the opportunity. It's good to talk.
Thanks. Thanks, Colin. I'd invite David now.
Thank you, Sinead. Kia ora tātou. Ko David Aitken tōku ingoa. I'm David Aitken, Interim Chief Exec at Lucidome Bio, and it's great to be here to talk about how methane vaccines can address some of the tough challenges we've heard about over the last 2 days. I'll first talk about Lucidome Bio and introduce the venture. Then I'll share some of our recent results, and I'll round out with a discussion on where to from here. Let's dive into the first part. So Lucidome Bio is a new venture launched in September this year to develop a safe, effective, and affordable vaccine to reduce methane emissions from ruminant animals. While the venture is new, we build on pioneering and long-standing research in New Zealand, led by Ag Research. And Lucidome Bio has a global vision to help farmers feed the world while protecting the planet. As we've heard, we're addressing a major global climate challenge. There's roughly 4 billion farmed ruminants globally, the majority on pasture-based farming systems, as we see in New Zealand. And the methane from the enteric fermentation that these ruminants undergo is responsible for almost 3 billion tonnes of CO2 equivalent emissions each year. It's roughly 5% of global greenhouse gas emissions. A methane vaccine can make a real dent on these emissions and help us to rapidly mitigate climate change. So how does it work? Well, a methane vaccine produces antibodies in the saliva of ruminants that's transported into the rumen. These antibodies bind onto targets on the surface of methanogens in the rumen, inhibiting their growth and reducing the amount of methane that the animal produces. We're validating key components of our prototype vaccine. Antigens are a key area for us. We've identified 385 potential antigens. We've prioritized down to 33 target antigens and are now validating the performance of those target antigens. We've identified adjuvant options to work alongside those antigens and are now validating the performance of those adjuvants. We're also exploring different vaccination strategies to improve the performance of a vaccine. So having introduced Lucidome Bio, how's it going? Well, over the next 3 slides, I'll share some of our recent results, starting with our antibody responses. So we know that our antigens induce strong antibody responses. So what you see here is a graph of titer. Titer is a measure of the strength of antibody responses. High is good. And so what you see in the orange line is results from one of our lead antigen formulations showing strong antibody responses relative to the control in blue along the bottom. We also know that these antibodies interact with methanogens. So these antibodies were tested against a cultured methanogen. We measured the antibody interaction with methanogen cells using a method that we've developed. And so what you see there in the orange bars of vaccinated animals are strong antibody responses and high antibody interaction with methanogens. So 1 is the maximum here, so we're seeing high antibody interaction relative to the control again in blue. These antibodies also reduce methane formation in vitro, so these are results from a short-term assay of antibodies against rumen contents. Antibodies reduced the rate of methane production in vitro, so you're seeing that in the orange bars relative to the green bars in blue. And this antigen formulation is progressing to cattle trials to measure methane in respiration chambers. So we're really excited about the results that we're getting. Where to from here? Well, speed and scale are core to our approach. It informs our technology development plans. There's various options for developing a vaccine. We're focused on a protein subunit vaccine as a scalable, affordable approach to develop and manufacture a vaccine. We're validating key components and, importantly, the assumptions that underpin our work. And we're working with partners around the world to help us move further and faster. We have an incredible team. Our scientists have pioneered methane vaccine research and development. We have expertise across key technical domains, and we're advised by leading scientific and commercial experts, including Professor Paul Wood, the chair of our advisory panel, Dr. Sinead Leahy, and others. We often get asked about timeframes. So our focus over the next 2 years is on demonstrating the key elements for a commercial vaccine: efficacy, duration of response, safety, vaccination schedule. And within 5 years, we want that regulatory approval in place to get a vaccine into the hands of farmers to deliver on our vision. We're very grateful to our research partners and our current and past funders. We couldn't do this work without them. They provide fantastic expertise, support, insights. We're working with them around the regulatory process and other elements of what we're doing. And we invite you to join us on this journey towards developing a breakthrough climate solution. I'd love to hear your questions, and I'm very open to the discussion that follows. Thank you.
Thanks, David. Technically, we're meant to go for morning tea, but we've got some time. So I'm just looking here. Yeah, great. I'm going to take advantage and we can ask some of the questions. You will have an opportunity after our next 4 speakers. We'll be bringing all of our panel members back up. You will have an opportunity to ask more questions then. But I might as well get into it now. Tom, obviously your solution is getting close to market, so quite a lot of questions from the audience. Any productivity impacts? Basically, how did you measure cows in the Waikato? And really around cost of bolus and dose frequency.
Start with a few easy ones.
Yeah.
Start with a few easy ones. So I'll second one first with green feeds. So the trial had green feeds out in the paddock at a herd level. So really high engagement with that. Really comfortable in the data. Productivity, it's good commentary on that this morning. We certainly don't have any conclusive data at this stage around productivity. A couple of reasons for that. One, our core focus has been on the core problem, which is can we get high duration of efficacy and high efficacy and do it safely. As our program now rolls out over the next 12 months, we'll have more of a focus on understanding productivity from our perspective. And hopefully we'll generate some conclusive data around that over the next 12 months. Ultimately, two different things, cost and price, right? But the price for a farmer to use the intervention at the farm has to be linked to the value that it creates. We're going to do the work to make sure we have really high quality verification of the methane emissions reduction at the farm in the environment, so that farmers can actually value get fair value for the emissions that they reduce. From a cost perspective, we're really encouraged with what we look like we're going to be able to manufacture the product at, and that'll come down over time, obviously as we scale. So we think the unit economics are really favourable in our certainly on our side at the moment. As we run the market development program over the next 12 months, we'll get more answers around price, and we'll be much more engaged with farmers and the value chain around what that looks like at that point.
Thanks. There's a couple of questions. It's really around consumers, and I think obviously what's happened with Bovaer. So people are asking, have you actually gone out? And this is probably for all of the panel members. Have you gone out and spoke to farmers or spoke to consumers to get an indication of whether your solution will be appealing to them? And maybe we'll start with the vaccine people first, and then we'll come back to you, Tom. So, Colin, do you want to start, and then we'll hand over to David.
Yeah, is that on? Yeah. We've had some good engagement with, mainly in the US, with National Cattlemen's Beef Association, DFA, etc. And so there's just a comfort with vaccination that comes from if you're assuring them that you're going to do nothing outside of the traditional animal practice, it's relatively easy to see adoption at that level. And that isn't us directly engaging with consumers, obviously, but it's a comfort level that they have with vaccination that we're comfortable with, leads us to belief that they will.
David?
For farmers, vaccines are an accepted farm practice. They already use it for animal health. Very much echoing what Colin said. I think if farmers know that there's a safe, effective, affordable vaccine that fits with their existing farm practices, that's something that they're keen to adopt. They're looking for these tools. That's what we hear from farmers that we speak to and from the partners that we work with. They want a tool that's easy to adopt and that they know will work. So I think there's a real demand out there for this sort of solution and a keen interest in what the vaccine ventures are developing.
I'd agree with those two things, and I'd throw on top of that, adoption is going to take way longer than we all want or hope, not just as a company but as an industry, because it has with every other technology that we've brought to agriculture. As I said in my introduction, I spent a lot of my time in the GM crop space, and even some of those solutions that were just substantially better for the environment economically. From a husbandry perspective, took a decade to adopt. So it'll take longer than we all want. The only other thing I'd add is, we have a commercial team in place now, business development manager on the ground in Australia, working closely with what we would call the big end of town, so the big processors, big producers, big retailers. And overwhelmingly, where the feedback we're getting is, how quickly can we go here? How can we work together and get this in market? Because there is demand there. So I think things like over the weekend will happen. They'll continue to happen. we shouldn't say that is the voice of the consumer. That is a voice of a representative group of the consumer that has a particular perspective. It'd just be like us as an industry going out and saying we've got everyone on side and we're going to go really fast here. That's not the case. There's a spectrum there. I think Beef and Lamb said that yesterday, and I thought it was really well said. So we're going to have those rocky moments along the way with interest groups. The core problem underlying all of this, though, is the planet doesn't really care about our preferences. It's getting hotter. And so I think if we're driven by, bring solutions to market that are going to make a difference, we're going to be okay.
Cool, thanks. I'll do one more question just for the two vaccine groups. Once you have a vaccine ready, do you foresee any main barriers to getting it out on the farm, or barriers to adoption that you might anticipate, whether regularly, consumer, etc.? David, do you want to start on this, and then I'll hand over to Colin?
I think there's a number of hurdles to jump over. Any solution will need a channel to market, an offer that's compelling to the farmers. We've had quite a bit of discussion over the last couple of days around insetting, offsetting, how some of this work can be paid for, particularly at a time when farmers are, feeling the squeeze. So, I think that business model, the revenue model, both in terms of the channel to farmers and then how that revenue is both shared and allocated out between the different stakeholders is a really key part. People need to be comfortable that the solution is delivering on its promise, so that after-sales support and these sorts of elements are, as always, important to the adoption of any solution. That feeds into business model decisions for any venture and what's core business to the venture and what can come from partners that may already have some of those relationships, some of that scale, some of that experience in other areas. So that's a key area for us, knowing what we do really well and working with partners that can fill the gaps in what we have.
Colin, do you have anything to add?
Yeah, I'll concur with that. I'd just add that what's unique about a vaccine is it's such a good logistics solution for the world, in that the quantity of antigen that you develop and put into a vaccine is in the milligram quantities. So we're not feeding grams per day or thousands of tons or millions of tons per year if you're in the seaweed basis. So we can go out through existing manufacturing assets, existing distribution channels tomorrow if we had a vaccine that worked. And I'd also go one step further and say it'll be the safest vaccine you've ever seen, because animals, cattle, ruminants have seen challenges to their bloodstream from methanogens through the course of their life. If you go looking for methanogen-binding antibodies in the blood of ruminants, you'll find them because they've seen it before. So it's not even like we're developing a new vaccine. We're going to be using existing components, and it's going to function in a way that is completely benign to the animal.
Cool. So with that, I'd like to thank again all of our speakers and invite you to go and have a nice cup of tea or coffee. Thanks.